Let me get started this seminar. Welcome to my seminar today. And good afternoon, ladies and gentlemen. I used to be a researcher in BLI. I worked with Dr. Matt Brenner over there and Dr. Jungping Chen. And I moved to Korea in 2010 and become a professor. Now I'm a head of department. So it's really thanks to everybody in here. So today my talk is in vivo real-time monitoring of mesothelial reaction in lung after cryo-injury. I'm going to show you some slide, and I think I can make it short. And here is my collaborator, Cholo Ok, over there. I accompany with him today. He is a pulmonologist in Kosin University, and I'm in biomedical engineering in Pugyong National University in Busan. So I will see you some picture of our school. Yeah, this is my school. In Busan area we can see snow usually, but last year we have big snow. It's really fun. And this is Kosin University College of Medicine, for him. And we two institutes apart, like 30 minutes driving distance. So we used to go and come back. And I have two prong researches. One of them is medical related research topic. I have some tools like optical coherence tomography. I think some of you are familiar with this topic. And I have been developed some endoscopy for OCT. And I'm interested. I have an ultrasound machine too. And he has some lasers for photodynamic therapy and laser ablation, and cryoablation and brachytherapy too. So we have some target disease like asthma and conjunctiva tumor and airway tumor, oral cancer, skin cancer, and OCT is kind of a diagnostic tool for some disease. And recently we are interested in asbestos-related disease such as mesothelioma. And another topic is related to engineering side. And I have some tools like Doppler optical coherence tomography. Optical coherence tomography has a Doppler function, so we can measure a velocity of some scattering particle. And also, I have a velocity measurement tool like particle imaging velocimetry, and of course, Doppler ultrasound and conductance probe. And I'm interested in microfluidics and micromixer analysis using those tools. And I'm also interested in boiling and condensation phenomena. It's a totally different story, but I'm interested in that topic too. We have a research team. 3 institutes had a MOU last summer. So in my department, we have 4 faculty. And then his medical center, almost 5 departments joined our MOU. And I have also collaboration with Gyeongsang National University, which has a veterinary medicine and animal medical center. So from last summer, we did a lot of work together. So this is my lab member. I brought some of my students over there, and I have 2 master students and 5 seniors, and I have some fund from Korean government. Okay, let's move into the main topic. Actually, this study was done because we are interested in asbestos-related lung disease, so-called mesothelioma. In developing country, so asbestos is banned right now, but long time ago we used to use asbestos. So that caused a disease in lung flora. So we are interested in this disease and try to diagnose in early stage. So, but we don't know much about the early stage carcinogenesis, so-called mesothelial cell reaction. I'm going to tell you what it is later. And no image-based study have been done yet. So OCT, I think, is the first, first experiment, which is an image-based study. And I used the OCT, and I made the inflammation using cryoengineering, which mimic the chronic inflammation caused by asbestos. And I'm going to show you some results and discussion and conclusion. I'm going to let you know what is mesothelium. In human body, we have several the organs which have mesothelium. First one is in your lung. We have a parietal flora and visceral flora. Those are mesothelium. And in your here, the parietal peritoneum and visceral peritoneum is another thing. And in male, this is male reproductive organ, and parietal layer of the tunica vaginalis and visceral layer of tunica vaginalis. That is another thing. Also, your heart has mesothelium. And we are interested in the parietal and visceral flora for this disease. And as you know, the OCT cannot penetrate more than 1 millimeter. So we have to develop some animal window, and we can let us see the inside the tissue. Flora itself is very thin, but it is buried by a thick tissue over there. So we have developed a thoracic window here, and the peritoneum window, and the scrotum window. And then this is the OCT image. Top part is parietal side, flora, and visceral side, and alveolar together. But in peritoneum case, the visceral side is very far from the parietal side, so we cannot image both at the same time. So I think this model wasn't so good. But in scrotum window, we can image both flora, both layer at the same time. So this model is where the disease happen, but we cannot access flora very easily. So that's why we developed the scrotum window instead of that one. But in this study, I'm going to show you some results with this window model. Actually, I'm going to show you some results using the scrotum window. And this is OCT image, which show you the microstructure in your human scrotum, the rabbit scrotum. So TS is testicular septal, and PL is parietal lamina here, and VL is visceral lamina at the down here, and ST is seminiferous tubule here, and scale by is about 1 millimeter. This is a 3D image of microtissue. And we confirmed the structure of tunica vaginalis, and his PL is a parietal layer, and VL is a visceral layer. And let's move on to the mesothelioma caused by the chronic mesothelial exposure to asbestos. So it causes mesothelioma. And currently, we have some diagnostic tool like a CT and PET and thoracoscopy. Thoracoscopy is like a gold standard in this area. But a CT or PET has false negative results. So we need to develop the other modality to diagnose better. So biomarker or optical imaging could be the solution. So OCT is kind of optical imaging. So and And especially in this study, I'm interested in the mechanism of early stage carcinogenesis. So that is related to the acute mesothelial inflammation caused by the cryo injury. So hopefully, I want to see the pathologic pathway to cancer. Yeah, this is a PET-CT result. Using this image from PET-CT, nothing wrong here. So it gives us false negative. But if you see the thoracoscopy result, then we can see the pleural tumor. And this result is a preliminary result I have done in BLI with Dr. Brenner and my friend here, maybe Jun and Ringfeng or Gangjun. And yeah, this is OCT probe with long green lens. Long green lens is just nothing but just a lay lens. But this probe is operated in the contact mode. So this is a pleural tumor developed by maybe Tanya or Dr. Brenner. Okay, so we contact the OCT probe on top of it, and then we imaged this tumor in 3D dimension, and we compare the tumor and OCT case in 3D OCT image, 2D OCT image, and pathology case. But as I told before, I'm interested in the early stage of the mesothelioma. So we mimic that inflammation using cryo injury. Well, cryo injury has a good thing in that it can cause 3 or 4 millimeter injury or inflammation in depth. So as you know, the foreign substance cannot be inserted into the pleural cavity without any hole. So that's very difficult, and chronic disease, is very difficult for us to mimic. So we developed a cryo injury model, and no report has been released so far with a cryo injury related to the pleura. So, and I'm going to define what is a mesothelial cell reaction. Mesothelial cell play a role in regeneration of the injured site. So mesothelial cell share characteristics with the stem cell in that proliferation and phenotype change are triggered by inflammation. So that is so-called mesothelial cell reaction. And here is the typical pleura. It is about several hundred micron in thickness. So OCT is a very good tool to image this pleura. But as I told before, it is in very deep tissue. So maybe photoacoustic tomography is another issue. So and anyway, OCT is located in here in the graph of penetration depth versus resolution. It has a resolution about 5 micron, and penetration depth is about 1 millimeter. I think most of them are familiar with the OCT principle, but some of the students is here. So I will briefly tell you some principle. So this is a spectrometer-based OCT system, and here is the row coherence source, and it's divided by 2, and one goes to reference mirror and reflect back, and one to the scattering surface in our sample. Here is the sample location, and then 2 light from 2 scattering and gives us some fringe pattern in a spectrometer. Spectrometer is consisted of grating, lens, camera, And so we have many scattering surfaces along the depth in our sample. So a scattering surface at the deeper position along G makes a high modulation in camera domain. So if we have a higher modulation, then it means we have a scattering surface at the deeper position in our sample. Then we do the Fourier transform from here to K domain to depth domain. Then it restores scattering event. And then if we scan our sample arm, then we can construct 3D image with a few seconds. So I developed an 850 nanometer system with maybe 100 nanometer full width at half maximum. So this is the characteristics of my system. Laboratory made central wavelength is 850. Spatial resolution is around 4 micron. And sensitivity 103 dB. Roll-off is a little bit big, 12 dB per millimeter. And I have a 70 kilohertz camera. So this is a thoracic window between third and fourth rib here. So here you can see the parietal side of flora. And we position the OCT scanner over it, and we apply the cryo probe too. So the temperature of cryo probe was maintained at negative 80 degrees Celsius for 30 seconds, and thawed for 30 seconds again. And we repeat this one 3 times. And oxygen saturation was monitored, and some anesthesia was done for animal. This is a New Zealand wild rabbit. So here is the OCT image through the thoracic window. The top layer is the parietal flora, and the black line is the parietal cavity, the pleural cavity. And the bottom white line is a visceral layer of flora, and A means alveolus. So we can see many structures at the same time because we used the thoracic window. And then we apply the cryo on it, and after 5 minutes after, we found some thickening in the parietal flora. So we're very curious about it. And then after 20 minutes and 30 minutes, it grows. So we have to see pathology to know what it is. Yeah, this is a live image of flora. So, yeah, and 2 days later, we took out the tissue, and then this is the window, and then we imaged the dead tissue, excised tissue again with OCT, and here is the focal thickening caused by the cryo injury. This is, and then we found another thickening around the site, and this was triggered by focal thickening, and then we found about what it is using the pathology. This is a kind of a proliferation of the mesothelial cell in the parietal flora. And this is a enlarged picture showing that this is a focal proliferation of mesothelial cell, and this is diffuse proliferation of mesothelial cell triggered by the focal point. And this is a visceral side of parietal flora. And then this is OCT, and then this is pathology researching. This is enlarged picture around here. So if you compare this image to the normal case, so OCT shows a very thick layer for a visceral layer. And then we can see some different shape of epithelial cells. So I'm going to compare these two. This is normal pathology, and this is the injured site one. So after comparing that one, we decide to make a mosaic pathology. So this is the injured area, and the cell shape is changed from flat to cuboidal shape. So it's like a phenotype changing. So it's called so-called mesothelial cell reaction. So two things. One is a proliferation. Another thing is phenotype change. It's confirmed by OCT and pathology. That's the result. So I'm going to conclude my talk today. OCT had a very good advantage in we can show some change in tissue in real time. So this is real-time monitoring technique. So we saw some change in pleura, which is a mesothelial cell reaction, but it is started as early as 5 minutes. And then this is a summary chart. We did OCT and histology, and I can divide in two groups for the parietal pleura and visceral pleura. And we have different time. This is in 30 minutes. This is almost immediate. And then after 2 days. And in 30 minutes, we can see a focal thickening using OCT imaging. And then in 2 days later, it is the diffuse thickening around the site was triggered by focal thickening here. And then histology gives us some the confirmed information. The focal thickening is confirmed as acute mesothelial cell reaction and focal mesothelial proliferation. And diffuse thickening was confirmed as a chronic mesothelial cell reaction and diffuse mesothelial proliferation. And histology shows us some cell shape change from small flat cell to tall cuboidal cell. So that's the summary chart. I think, this is acknowledgement. This work was done by several undergraduate students, actually. So I'm very proud of they're over there. And then some very helpful advice from the several the doctors, from pathologists and occupational health experts and chest surgeon, and Choro Og is over there, and he is interventional pulmonologist. This work was done by the 3 institutes under the same MOU. Okay. Thank you for your kind attention. Do you have any question and comment?
So are you planning to use some of the depth-dependent reflectance data to get more characterization of the cell characteristics, maybe cell shape or size,
right.
density, those types of things?
Yeah, that's good advice, I think. Yeah, so we have to do it like that. Yeah, but so far we did OCT study only. Yeah, but actually, we're gonna do some detailed job like. We can insert some nanoparticle through the airway, and then it hopefully it causes some inflammation around the pleura. So we can get some live image with a nanoparticle injury. So yeah, that's another story to go, I think.
Does the histopathology show enhanced vasculature as well?
Oh, it's very early. So it's just the inflammation, so it's not, have any vasculature. Pardon me?
How do they currently diagnose? Same thing you're looking for.
Mhm.
How do they, as the current medical knowledge, how do they diagnose the same thing, like early stage cancer?
Early stage cancer. Yeah. I don't know, So yeah, OCT tried to early diagnosis, but is I have no idea about that yet. So I'll tell you later.
That's a big problem.
Yeah.
Are you thinking of using some of the spectral content also for other contrasts? Or will you, from a technology development, where do you see that going?
Actually, I'm interested in the photoacoustic tomography because that site is buried in very deep tissue. So OCT, limited in penetration depth. So we have to make a hole anyway to diagnose it. That's not very good thing. So, but in thoracoscopy is a gold standard. So OCT can be integrated to the thoracoscopy, but it is kind of an invasive method. So photoacoustic tomography can give us a non-invasive method, hopefully. So that's another direction to go. Yeah. Okay. That's it. Thank you.